I'd like to introduce Dr. Raheli Kafir.
Hey, hi everyone. Until it opens. As you know, my name is Rahele, and it's great to meet you over here. I'm just going to talk about application of artificial intelligence in bioengineering here. And to start, as we know, in the 20th century, there are lots of technologies and information from physics were combined and brought us some things like our mobile phones that we cannot live without them anymore. And it seems that a combination of bioengineering and AI these days is something very similar to that. And we will experience a lot of improvement in next years. Talking about the biological systems, they are usually very challenging to be analyzed because of many issues like their complex nature and also the range of data. They may be very simple and very complicated to be analyzed. And also the curse of dimensionality. We need actually a lot of data to prove that our algorithms are accurate and acceptable. But the problem is that we usually do not have enough data in biology. So all of these provide us opportunity to think about application of machine learning and artificial intelligence to see what they are doing. We want to have a kind of introduction to say machine learning is the capacity of the machine to somehow do and think exactly the way that the human does. And machine learning is shown to be able to solve some problems and find some patterns that are somehow very difficult to be found in biological systems. Before going into the next slide, I just want to talk about two main types of machine learning and their differences. We know that there are some methods known as supervised and some as unsupervised. There are some new methods in between. I don't want to discuss about them. In supervised methods of machine learning or artificial intelligence, we need a lot of data that's labeled and we know how they are categorized. Using those information, we train a system, and after training, we have one that works on unseen data. But in case that we are talking about unsupervised methods, we do not show the algorithm what is correct or not. An algorithm tries to find the patterns and find the groups that are better for discrimination of the data. Both methods and similar methods are shown to be able to solve lots of problems. For example, for diagnostic, prognostic, and predicting the response of treatment through medical data. And also, as it was presented in previous slides, you saw that we need some localization and tracking issues, for example, finding targets which are moving in the videos, and we can use them for image-guided therapy or so. And also design and development of drugs, proteins, DNAs, and lots of similar approaches, and solving problems in biomechanics and biomaterial. These are approaches that are already used in the area of machine learning. But the reality is that AI is not a solution to all of our problems. There are lots of challenges. We should be aware of them and try to avoid them or somehow solve them during our methods. One of the well-known parts of computing power. We need a lot of computing power to be able to analyze them. We need the transparency and interpretability in order to build trust. I mean, for example, if I want to say clinician that I am able to say if this image is coming from patient or a healthy control, we somehow should persuade them to say, yeah, this is not a random answer. We need some interpretation that we will talk about. The data privacy and ethics is really important because we're talking about healthcare data. And the bias problem is, for example, when you have data bias, you train and test your algorithms on a set of data in a local hospital, and you want to say, yeah, it's working, but it's not working. It's biased to a special part of data, not for all of the data in the world. And finally, the problem of having enough data, as we discussed, and also problem of capturing expert knowledge is very complicated because it is really difficult to want a clinician with busy clinician to sit down and categorize or mark some labels for you. So we should consider all of these. I try to have some Christmas tree here. It's a kind of road to say what is my research interest. It's based on artificial intelligence during last 7 years, I can say. But two other things that I'm really interested in, time frequency analysis. I mean, looking into signals and images from both sides of time and frequency and considering them together with wavelets or so. And also graph theory is one that I'm really interested in, modeling our programs, modeling our biological system or data using graphs and applying graph theory to solve our problems. And I tried to somehow combine them together, and it seems that they're working good in combination. But talking about the application of AI in bio-related works, I should talk about bioimaging and biomedical signals that I've already worked. As you can see from this plot, majority of my works are on eye. But just to discuss about the other parts, I would like to talk about some of my works on brain. For example, on fMRI or MRI data, or EEG data coming from different diseases and different people with healthy background to say who is who, what's the problem in them. Using thermal imaging in order to early diagnosis of cancers in breast, and also some works on COVID-19 data on CT and X-ray images. And one of these were one of our mostly highly cited papers that got around 600 citations in 2 years. Some of our works were on exactly again in the COVID area to say how many people will suffer from COVID if the government make different decisions. If they close the schools, what will happen, or otherwise, what's the reason? And I had some works on heart, as was presented in previous slides, to localize them, to use the signal from heart to diagnose different diseases. And well, I have two brothers, both of them are dentists, so I had a lot of data from dental background. So I had some works in this area, and some 3D reconstruction and localization and so from CT images of the pancreas. But in order to concentrate on what I am mostly interested, ocular images are the ones that I've worked on them for many years. They are usually in 3D or 2D and different movies or so. I am mostly interested in application of ocular data in diagnosis of neurodegenerative or NDD diseases. And these are some diseases that are common between brain and eye, like multiple sclerosis, Alzheimer's, Parkinson, and so. And the problem is that usually the changes in brain are very difficult to detect, and you may see the symptoms of the patient before you can diagnose it using their brain. So the clinicians, the neurologists, are looking for a way to detect NDDs in their very early stage. And considering the eye, we know that the retina is somehow an outgrowth of our brain. They have the same environment. So retina is the only part of central nervous system that you can see directly from outside, and ocular imaging is exactly the way that we can see it by 3D or 2D data. This 3D data that I'm mostly interested in is called optical coherence tomography, or OCT. It gives information about the layers of retina and very high resolution cross-sectional images. Well, having a look in timeline of works that I had on ocular images starting from 2010 to now, you can see these yellow circles showing the application of machine learning in this area, and you can see that it's increasing as we come into these years, and it's very popular, as you know, for other applications. Just to have a kind of journey to say what I have done, I selected a few of them, all connected to each other with a special purpose, and I want to go through this story. Well, when we talk about diseases that are available in eye, I mean ophthalmologic diseases, as you can see from this image, the changes are so obvious. So me or you, as non-experts, can say that that one is normal, the other ones have some bumps or so, that's not normal. So in those cases, for ophthalmologic diseases, it's not that complicated to send these images, I mean raw images, into our algorithms and expect that they produce some results for us. I mean, the network or model can think exactly as we do easily and decides. We had some publications, I tried to mention them in lower part of the slides. However, the problem comes when we concentrate on NDDs, as we talked about different diseases that affect both eye and brain. And it is very challenging to see that there is very small and subtle differences between the eyes with different diseases of NDD and normal eyes. So it's not that easy, even for an expert, to look at them and say if they're coming from patient or a disease. one solution to this problem is just concentrate on very small information and extract the most informative part of the data. For example, by finding the exact location of cells inside the retina by a process that is called segmentation. I mean, finding where is each pixel located, how much is the thickness of the cells, and how much of cell do we have in each layer, and we expect it reduces with some problems like Alzheimer's or so. However, the process of segmentation, particularly if it should be done manually, is really time-consuming, prone to error, and has lots of problems. So we need a kind of process to find it automatically for us. But you remember we talked about supervised algorithms? We usually need a bunch of data to show our algorithm and say, yeah, this is the correct answer, and our algorithm wants to look at them, learn them, and then mimic them for us. So how do we prepare those labeled data? We should request the clinician to sit, a busy clinician that doesn't have any time, and do click, 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 lots of clicks on each image, and it takes a lot of time. So we developed a method based on graph theory in which it needs only a few clicks and just finds the location of mouse and calculates the best path until the next pixel, and finds it somehow in a semi-automatic way, finds those labels for us. So this is trying to solve this challenge, the difficulty of capturing expert knowledge. And then we designed a kind of method for automatic segmentation using lots of new modalities, like, for example, how we decide about the cells. We see that there is difference in their textures, so we give it as a kind of cheating into our algorithm to be aware of those and do the segmentation for us. And finally, as you can see in this publication, in the last column, the results are really good. And we tried to show that it is working on very different data sets coming from different countries, different devices, and people with different problems like Alzheimer, Parkinson, or so, and it's working perfectly on all of them. Well, the segmentation is done, and we want to send some useful information to our classification approaches to say if some image from the eye is coming from a patient or a normal person. We need, again, to extract some useful information, as I told you, about the amount of cells in each layer. So we prepared these 8 images as thickness maps, showing the amount of cell in every area of the eye. And we use different classification methods. I just want to say the last one that we used, a kind of deep learning method, using those images as input. And the most important part was to provide us a kind of interpretability. I mean, we wanted to show the clinician, I mean the neurologist and ophthalmologist, which part of the image was considered by my network to decide correctly. If my network looks at the same area that the ophthalmologist would look in that case, it seems that it's thinking correctly. But if it looks outside somewhere else, it seems that your network isn't working correctly. So this kind of interpretation shows exactly the area that it is known clinically is important for example, multiple sclerosis that we were working here. So we provided somehow interpretability. And well, just to final slide, because we don't have a lot of time, we just want to say that I have lots of work to do on NDD project, as we talked about gathering different data sets from multimodal presences and so. And I also am interested in somehow other projects like AI-assisted patient triage system, in order to say who is most important and critical to be seen by a specialist in our NHS system, for example, and who can wait yet and has time. And thinking about image synthetic to fill the gaps that we usually have. I'm interested in gait analysis and progression analysis of Parkinson's disease, and I'm making new collaborations in department. The first of them is the one that we just started on endoscopic capsules, and I hope to continue. Thank you very much for your time.